Okay, can you see my presenter mode screen?
Yes.
All right. Well, I guess we get started then. Well, hello. It's nice to meet you all. Thank you for joining us today. My name is Janina Grumbeck. I'm the Director of Microbiome Applications here at MiDOG. By training, I'm not a veterinarian, but I have a PhD in microbiology and molecular genetics. And with that background, I have been always very fascinated how bacteria and fungi influence human and animal health, and have studied specifically gastrointestinal health for a couple of years in Lincoln, Nebraska during my PhD. And over the last couple of years, there have been more molecular technologies developed that allow us to study the interaction between bacteria and fungi and the animal and human host in greater detail. And today I'm going to present to you how these type of molecular analysis can help you with veterinary diagnostics as well, and how it can help um, treating and detecting infectious diseases in our companion animals much more efficiently. If you ever have any questions during the presentation, feel free to interrupt, or if you have um, any other items you want to bring up during the presentation, or if you want me to elaborate on any topics, please feel free just to interrupt me. So let's get started. Of course, as clinicians, you know how important it is to have an accurate pathogen identification in order to treat your patients the most successfully. You have to know whether you're dealing with a fungal or bacterial or viral infection in order to choose the appropriate antibiotics or antifungal. So accurate treatment always starts with accurate pathogen identification. However, there are so many pathogens out there that are incredibly difficult to diagnose, yet they are very frequently and commonly cause infections in our pets. And those organisms include Mycoplasma, Mycobacterium, Fusarium, or some of those fungi like Naniziopsis. And some of these organisms don't grow in the lab at all. Others can take several weeks to grow, like the fungi. And so that is a lot of time spent waiting for you and your patient. You might even have to consider empirical treatments while you're waiting for the results to come back. And this kind of diagnostics approach has left us with a considerable amount of challenges because at this point we have millions of animals suffering every year from often multifactorial infections. So these infections include two types of bacteria, often a streptococcus and a staphylococcus, or a bacterium and a fungus, or sometimes even a viral infection as well. And for every single antibiotic that's currently on the market, there are already pathogens that have been isolated from patients that are resistant to them. So those would be MARS A or MARS P. And those pathogens are so severe and so resistant against every single antibiotic that we have currently available, even in human diagnostics, that by 2050, the leading cause of death will be from microbial resistance infections. So that has really inspired the One Health approach, the judicious use of antibiotics, which also influences veterinary diagnostics as well. And it is indeed time to act now because 2050 is not that far away at this point. The still current standard of infectious disease diagnostics would be culture and sensitivity testing. It is still considered the gold standard, maybe because it was developed during the golden age of microbiology, which was about 180 years ago in the 1880s, developed by Robert Koch and Louis Pasteur and Jonathan Graham. And that is the time when gram staining, petri dishes, and those types of technologies were developed. And if Robert Koch would walk into a microbiology lab nowadays, I think he would still feel right at home because while some of the media that veterinarians have or microbiologists have developed have changed and we have more automated plating and things like that, the principle has remained the same. You still need a variety of different agar plates in order to identify all those different types of organisms that might be causing the infection in your patient. So including different temperatures, different oxygen levels, different water content, different vitamins, different carbohydrate sources, etc. So you often need multiple tests to target different types of organisms. The turnaround time really depends on which type of organisms you're testing for and looking for, and can be between 2 to 7 days. For fungi, it can take several weeks, and as I mentioned previously, some organisms do not grow in the lab at all. The great benefit is, though, that you have the ability to test for phenotypic antimicrobial resistances and sensitivities for those different type of antibiotics you have available, and you can develop the MIC. However, there are a few limitations to consider with this type of approach, and there are those quick growers like E. coli that doubles every 20 minutes, and the space on the petri dish is quite limited. So once E. coli really starts proliferating, it might overtake other bacteria that are causing the infection in your patients, like Mycobacterium that grows incredibly slow. So the identification of those slow growers might be inhibited, which can have a huge clinical significance for your patients because you are creating a very artificial setting in the laboratory. It's not the same setting as it was on your patient. So this artificial laboratory setting might just benefit some of those fast growers or those that we can easily grow in the lab. Then anaerobic organisms do require special testing. As soon as you take the sample from your patient, for example, from a urinary tract infection, and the sample is mixed with the oxygen in the air, some of those anaerobes just immediately might die off. And so you can't even grow them in the lab because they simply died off in transit. Once they do reach the lab, you need to have anaerobic chambers, which not every clinical testing lab is equipped with. If you're looking at exotics, they have a whole large number of special needs when it comes to infectious disease diagnostics. Most laboratories focus on mammalian temperature, so 37 degrees incubation temperature, which is great for dogs and cats, but can be a little troublesome if you're looking at a patient that is a bird or a reptile who have a different body temperature. Because of their different body types, they also often require uncommon media, and more often than cats and dogs, they suffer from fungal infections, which again take special incubation media and can take several weeks to grow. A study that came out by the AVMA in 2020 looked at the perfect case scenario of UTI patients. So they had dogs coming in with all signs of a UTI, blood in the urine, high amount of protein in there. They were staining while they were peeing, and they saw even rods and cocci under the microscope once they collected the urine via cysto. And they put the urine immediately into the incubators with the incubation media without the more common approach when it is in a clinic. You have to wait for the pickup service. The urine might end up in the fridge for maybe an hour or two before it gets taken to the lab. So they had the perfect scenario, immediately started putting the urine into the incubator, and still in 67% of those samples, the growth came back no growth results, even though they saw the rods and cocci under the microscope. So when your urinary tract infections culture growth come back no growth, you wouldn't be that surprised. For other sample types like swabs of the skin and ear, the no growth results are a little less common, but they still occur in 10 to 30% of the cases, which is quite considerable and is a pain when you have to explain that to your clients and for you as a clinician in order to make your treatment decisions. And if you consider how many microbes live on this planet, it's about 10 to the 20, an incredibly large number, and nothing in this world is sterile. Even urine from clinically healthy individuals is never sterile. But cultural reports come back often with no growth. And when you look at specific studies that investigate how many cultures come back negative, it is between 1 and 30% of microbes that we can actually grow in the lab, which means that culture is missing between 70 and 99% of all microbes on this planet. Not all of them are infectious agents. There might be some commensals and environmental organisms, but still, it's a quite large number. So when your results come back no growth, it shouldn't be that surprising. Another issue are biofilms, which are entirely unculturable, and I will go into more details on why that is in some of my later slides, but yet they cause 80% of all infections. And another sensitivity of culture testing is that you can get the sensitivity for only those organisms that actually grow. This whole phenomenon about the viable but not culturable has been around since the 1980s that we are aware of it, and that was part of the reasons why PCR and other molecular technologies have been more implemented into infectious disease diagnostics. So to briefly summarize, there's 10 to the 30 microbes on this planet. Only 1% of them are culturable, which is referred to as the viable but non-culturable phenomenon. The alternative is the molecular technique of PCR or quantitative qPCR testing. The benefit is that it's completely growth independent because the DNA or the RNA of those microbes is used as the analyte. And with that, it has a very high sensitivity and specificity towards the target that you select. You can get results really fast, especially if you have a machine in-house. Sometimes you can get the results even within 2 hours. And if you're lucky enough to be able to select qPCR, your results are even quantitative. So rather than just saying detected, not detected, positive, negative, it can give you a quantified number just like culture would. Normally, you're limited between 8 to 38 targets, and the benefit is you will get an answer for those targets that you selected. A limitation, however, is you will also only get an answer for the targets that you did select. So the panel quite often returns all negative, depending on how well your educated guess was. But there might be more uncommon and more unusual pathogens that are infecting your patient. And when the panel returns all negative, you find yourself having to select a secondary panel. Again, more time and money wasted while your patient is waiting for the results. There is also a relatively high rate of false positives, which can lead to misdiagnostics because this approach has a high sensitivity. Some panels do offer antimicrobial resistances, normally between 10 to 15 gene targets are available in those panels. A consideration when you're looking at these panels is that you're using the genotypic antimicrobial resistances. So unlike culture where you grow the organism in the lab and you can test those antibiotics directly on them, here you're using the genetic potential, the genes present in those bacteria, whether or not they will be resistant against an antibiotic. In some cases, that can even be more sensitive than culture because, again, it's growth independent, so you don't rely on actually growing the organisms. You test the genetic potential, and in that way, it can be more sensitive than culture. There is a novel alternative that combines the benefits of both worlds, and it's called next generation DNA sequencing, or short NGS-based diagnostics. Just like PCR, it uses the DNA or the RNA of those bacteria and fungi as the analyte, but unlike PCR, it targets very broadly all bacteria and fungi. So there's no panel selection needed. PCR usually requires either genus or more often species-specific primers, so only looking very specifically at a very small number of targets. This approach looks very broadly at literally all bacteria and all fungi. And how that is achieved is by looking at the 16S RNA gene of bacteria, which all bacteria have, but it's still specific enough to each bacterium that it can go down to the species level. So similar to a thumbprint, all of us have one, but it's so specific with the crips and curves that we can identify an individual just by looking at the specific design of each foot thumbprint. So the same applies for 16S sequencing for bacteria, and for fungi, you target the ITS region. And with that, you do not have to select any panels, but you have the same high sensitivity and specificity that you have with PCR testing. Like qPCR testing, it offers quantitative enumeration of those bacteria and fungi that are present, similar as culture testing, but it also allows for novel pathogen discovery because you're not limiting yourself in any way by specific targets and you're looking so broadly. You might be able to discover novel pathogens that are causing the infection in these patients, even though they were not on your radar previously. It also allows for microbiome analysis, and what that means and what that can offer you as a clinician, I will go into more detail later as well. A limitation is that this workflow is very complex. It cannot be done in-house. It requires a lot of high-skilled technicians and machine equipment, and therefore it has to be shipped out to a specialty lab. A consideration is the same as for PCR testing is that you're using the genotypic information for the antibiotic resistance profiles. Again, you're not growing any of these organisms, you're looking at their genetic potential. So how does this NGS testing work? So for you as a clinician, it's just as simple. Return to them. Oh, Mel, can you accept those people waiting in the lobby, please? Okay, so when you collect a sample from your patient, you have to ensure that you really grab all the organisms that are present and are causing the infection, not those that just happen to grow in transit. So whenever you collect a sample from a patient, you have a large variety of different organisms, gram-positive, gram-negative, aerobes and anaerobes, um, rods and cocci, and you need to ensure that you can actually preserve this microbiome profile until it's analyzed in the lab. And how you achieve that is by providing a DNA RNA preservation agent. So as soon as you mix your sample with this agent, it's stable at room temperature. Nothing will grow, nothing will decay. So it's a true snapshot of all the microorganisms that were present in the patient at the time of sample collection. With that, it doesn't need to be refrigerated and can be shipped to the lab. Once the sample is acquired in the lab, the DNA of all those microbial organisms has to be extracted. And it's very important to extract the DNA of all organisms present, not just those that give their DNA up fairly easily, like E. coli. You barely have to put any enzymes in there. E. coli immediately lyses and you have all the DNA. Other organisms like Pseudomonas aeruginosa or some fungi are significantly tougher to lyse. So you would use enzymes as well as bead beating to get the DNA of all those organisms out so you have an unbiased view of all those that were present in the patient. Once you have the DNA, you have to purify the DNA similar to PCR approach, remove PCR inhibitors, you mark the DNA, you put primers on it to start sequencing. And most sequencing is done using Illumina, which are these tabletop machines that are about the size of a large luggage. They look kind of fancy. They light up and have all different lights coming out of them. And they generate millions of DNA sequences. That takes about 20 hours. And you generate those DNA sequences of all the organisms, all pathogens, all commensals that were present in the sample. And at the end of the day, it's just combinations of ATCG over and over, about 200 reads of just ATCG in different combinations. And once you have those reads, you need to map them to a database. Similar like you would have a new word that you haven't heard of before, you would grab a dictionary to understand what does this word mean, what is the origin, how do I use it. This is the same approach that we use for these combinations of ATC genes. So we map those genes down to what kind of species is it? Is this a commensal? Is it a pathogen? What type of resistance genes do we have? And then to generate a report that only highlights those clinically relevant organisms rather than all those millions of DNA sequences that were reported. So it is very comprehensive and covers all pathogens because it is looking so broadly at all of them without any predisposed issues. And if you are familiar with the American Society of Microbiology, according to them, this type of testing has the potential to revolutionize clinical microbiology drastically because it is this single, all-inclusive diagnostic test. And if we just look at the body of literature that is coming out, specifically looking at NGS in clinical diagnostics and how it can be used, it really proves them right. NGS became publicly and commercially available in about 2005, and since then the number of publication has skyrocketed with just 5,000 publications last year. The majority of these publications is focusing on NGS diagnostics in human diagnostics, but veterinary medicine is rapidly catching up. We have at this point about 43 different peer-reviewed publications highlighting the utility of this type of testing, including novel pathogen discovery and comparing it also to culture and PCR testing, with a large variety of different types of bacteria and fungi being targeted, as well as different types of animals. As usual, dogs are the most common pets, so the majority of publications is focusing on them, both on their oral health as well as gastrointestinal and skin health. But cats are following up with sample types like oral health or UTIs, as well as some of the exotics. We at MiDOG also actively participate in this type of publications, and if you ever want to learn more about that, I'd be happy to talk about it. We have at this point 15 different peer-reviewed publications highlighting the utility of NGS in clinical diagnostics. One surprising fact that I couldn't quite understand why it hasn't been done yet was that there are so many studies looking specifically at sample types for culture testing and NGS, using the same sample and just splitting it in two and seeing what are the results, what are the benefits, what are the limitations of each technique. But no one has done that for small animals and exotic animals. So we set out to do our own study, and we did this right in front of the Exoticon. And within 2 months, we reached out to all of our clinics that have been working with us in the past. And in just 2 months, we got 72 samples from 13 different clinics. And the only inclusion criteria were that it is a patient that is either suffering from a confirmed or suspected infection. So it was not a wellness exam. And that a sample was collected at the same time for their traditional culture and sensitivity testing and for NGS testing, so not a week or a month apart. And then we just looked what kind of results we would find. And I'm biased. I will definitely say that I know molecular testing is more comprehensive than culture, but just by how many pathogens were missed by culture, I was a bit shocked as well. So here are the results, and this publication will be coming out shortly, so you can read up on all the details. We found that overall culture came back positive for any type of organism in 75% of the cases and in 98% of the cases for NGS testing, and culture detected 20% of those in pathogens. Keep in mind, those were samples from a suspected or confirmed infection, and still only 20% of those had pathogens present. NGS testing had pathogens detected in more than 55% of those cases. The other ones were what is traditionally more considered a commensal, but commensals can turn pathogenic, and I'll talk about that in a little bit later in the other slides as well. Fungal culture came back positive in 7 or 14% of the cases, depending on how you count. To be fair, only half the samples were actually tested for fungal testing. So if we assume that half of them were tested, 14% came back positive, versus NGS testing had almost 80% positive. And in half of those samples, we actually detected pathogens. And when I'm referring to pathogens, here are the specific pathogens we identified in this sample set. And they included traditional ones like Mycoplasma, Pseudomonas aeruginosa, Ureaplasma, Campylobacter, Corynebacterium, Mycobacterium, Chlamydia, and so on. And for the fungal, we were looking at bacteria and Anisiopsis, some of those that you traditionally even see in exotics. So to summarize our results, culture missed 65% of selected bacterial pathogens and 97% of fungal pathogens, and in even 34% of the cases, important fungal pathogens were missed. So this is hinting at a fact that fungal infections are much more dominant than we currently anticipate, simply because fungal culture is not done very commonly anymore, because quite often the results come back no growth anyways. And do you want the results 3 weeks later after the patient has already gone home? So it's quite understandable that culture testing for fungal infections is not commonly done anymore. But this is a good reminder that fungi are severe pathogens as well. So hopefully at this point I have convinced you that NGS reliably detects those pathogens, but also so much more. If we were to swap even our own clinically healthy skin just a little square centimeter or so, we would find about 250 different types of bacteria. It is an ecosystem that is often very highly diverse. So if you were to swap your patient, even though there is a clear infection, you might get some commensals as well. Is that simply too much information? You do not want 200 different organisms. What do you do with all that type of information? So The benefit of NGS testing is that it looks broadly at the entire ecosystem. It will detect, as this simplification of it, it will detect all those different types of trees, all the bunnies, all the deer, all the frogs, and all of that. But it will also detect the pathogens. And the clinical diagnostic lab will never just give you all this massive information. It will digest it for you and highlight, hey, we did find commensals, but hey, this is the pathogen we identified. In this example, the carnivore, it is one fox. There is no other wolf, just one fox. And so this was what you would focus your treatment decisions on. Simply compared to culture and sensitivity testing, where they would simply say, commensals detected, but here are the pathogens. An NGS diagnostic lab will also highlight those pathogens for you. But the other benefit is that it does look so broadly at the whole microbiome analysis and that can identify when a commensal even turns pathogenic. And what is the microbiome really? It became very popular in about 2015 or so. It even made the cover of the Times Magazine. You can now test your own stool, offered by a lot of variety of different companies, or your stool from your dogs and cats, looking at the microbiome and deducting health benefits from that. So the microbiome is from a Greek word meaning small life, and it encompasses the microorganisms living together in any given habitat. So that includes bacteria, fungi, viruses, protozoa, and all of those together. And if you were to guess, how many different microbes do you think live on a pretty large dog? Keep in mind there's 10 to the 30 on the planet Earth. And I'm just going to wait a few seconds if anyone would like to shout out their best guess. Okay, nobody? All right. Well, the answer is about 39 trillion, an incredibly large number, and the majority of those are living in the gastrointestinal tract. And in order to study how they influence the canine and feline health, we do need to use molecular technologies because keep in mind 99% of all those microbes are unculturable. But over the last couple of years, since NGS diagnostics and analysis has been developed, we've learned so much more what these bacteria can do for the animal health. They're involved in all aspects of health, from promotion of fat storage on how our metabolites are broken down, how we metabolite therapeutics and medication. They produce short chain fatty acids that keep our gastrointestinal lining really tight because the majority of those organisms do live in our GI tract, but we do not want them entering into our bloodstream and things like that. So they help with protection against epithelial injury, and they break down our food components as well as helping with resistance to pathogens. And by swabbing thousands and thousands of different individuals, we can start seeing that there is a strong signature of what a healthy microbiome looks like, whether it's the skin or ear or gastrointestinal tract. In most of the cases, it is characterized by a large variety of different organisms that are kind of at an equilibrium with one another. However, in an infectious disease case, we do see all those pathogens. We don't have to worry about the viable but unculturable approach. We don't have to worry about biofilms, which are unculturable. And it also identifies an imbalance. At what point does this healthy microbiome shift towards an infection? Because rarely an infection is an on-off switch, right? There's a pathogen entering the ecosystem, it's proliferating, it's slowly growing, and then until you have a full-on infection, it is a gradient. And by using the overall microbiome profile, you can start seeing at what point does the pathogen keep growing and at what point is it an infection. And after a treatment, at what point is the antibiotic working, the pathogen is going down, and I reestablish a healthy, diverse commensal microbiome. So using this type of technology can help you identify where is this imbalance, and on this imbalance scheme, are you going towards health or are you going towards disease? And we're learning so much more of how this microbiome influences health and what are the key players. Because in reality, even though there are 200, 300 different organisms on my skin, not all of them are key players in keeping a microbiome healthy or moving towards disease. And we've been studying this type of phenomenon for the last couple of years, and we're lucky enough to publish our work in veterinary microbiology 2 years ago. And for this, we swapped thousands of dogs that were either clinically healthy or asymptomatic or suffering from either otitis or dermatitis. And we saw that those dogs that were healthy had a large variety of different bacteria and different fungi in their ears and in their skin samples. So if we take all those different organisms we find and set that to 100%, that represents the entire donut here. And then if an organism is more abundant than others, then their donut piece would be a little smaller or larger. So in this case, every different color on this donut is a different bacterial species, and here different fungal species. And you can see there's a lot more different bacteria than there are fungi, and the same principle holds true for the skin. However, in a clinical infectious case, there is an imbalance, an overrepresentation of just one or two different bacteria. So you start having very strong signals between health and disease. And you can start learning and use artificial intelligence and machine learning algorithms to identify which of these organisms are indicators of health and which are those that are indicators of a disease state. We did a similar study for bunnies. Like I mentioned, dogs are leading the way, but the exotics are quickly catching up. And we did the same study with otitis, and we saw that the bacterial profile is still quite diverse, more diverse than the fungal profile, but not as diverse as compared to a dog sample. And that actually in the clinically healthy bunnies, this organism was the most dominant one, which is also the most dominant one in the otitis ones. But the secondary most abundant bacteria are then the indicators of health and disease. Here shown in orange is quite dominant here, but barely present in the otitis cases. And then the opposite here, this dark blue one, is quite dominant in the otitis ones, but barely present in the asymptomatic ones. So you can start using all this type of data to identify these novel organisms and identify markers of health and disease using this microbiome analysis. We're not the only ones working on this type of technology, and this is work that came out of Texas A&M, and they studied the microbiome of dogs and puppies from the nose to the tail. And on the left side, you see how diverse the microbiome for bacteria is, and on the right, how diverse it is for fungi. And the most important markers driving this diversity and which type of bacteria you will find on the skin or in the nose is the water content. So the mouth is a lot more moist than the skin, and then the nose is a little bit in between and things like that. So that is how these different microcosmi drive this different type of microbiome. But again, like we saw in our study as well, they saw strong signatures of health and disease. This is an example of canine allergic dermatitis. And in this case, we're looking at the fungal profile. And in the clinically healthy, there were these 12 fungi that were significantly more dominant in the healthy samples, and there's only 8 fungi that are significantly more abundant in the disease state. So instead of having to worry about these hundreds and hundreds of different fungi, it boils it down to only 8 pathogens and 12 markers of health. So that becomes much more digestible. Similar, if you get your own blood work done, you look just at a few markers of health and disease. You don't have to look at every single marker. So this is to highlight to you that even though there are hundreds and there are commensals detected, the microbiome lab will help you identify those markers of health and disease to help you with your diagnostics so you can make your decisions much more educated and much more quicker. We were able to form this whole pathobiome map on a clinical example of dogs with periodontitis. We had puppies that were well, not only puppies, also adult senior dogs that were either healthy and had perfectly healthy gums in group 0, and those that were suffering from severe periodontitis in group 4. And then we had groups in the middle, and we identified those markers of health and disease, not only which bacteria are present, but also how much of them. And it came down to only 39 markers. And so if you were to swap your dog's periodontal samples, you could put yourself on the map and see, is my pup more towards health or more towards disease? And after a treatment, am I moving on the gradient towards health? Hopefully, or do I need to have another intervention because I'm still progressing towards a periodontal state? And so this work was just published in the American Journal of Veterinary Research. So at this point, we know NGS identifies all those pathogens, it quantifies them, and it even helps with the discovery of novel markers of health and disease. But now that I have those information, how do I actually treat my patient? And this is where I would like to do a quick detour back to the microbiology classes, because we distinguish between two types of antibiotic resistances. The first one is intrinsic resistance, so the innate ability of a bacterial organism to resist an antibiotic. So antibiotic usually target different types of the life cycle of a bacterium in order to kill it. So that could be the Krebs cycle or breaking open the cell wall so those bacteria literally leak out. So if a bacterium is missing that specific target of an antibiotic, it will be resistant. For example, if it doesn't have a cell wall, the antibiotic is targeting a cell wall, it won't work. So this is all based on published data. It's always consistent for all given species. So all species of E. coli will have the same type of innate ability to resist an antibiotic. All this Staphylococcus intermediates will have the same resistant. So you basically can look it up in a database if the organism will be resistant to this antibiotic or not based on intrinsic resistance. On the other side, we have acquired resistances. So these are genetic mutations, and those are very specific to each individual and each individual sample and each individual bacterial cell. So a bacteria might have a mutation just because of the pressure of having an antibiotic present, and they double very quickly, so mutations happen quickly, and that's just how a bacterium might become resistant to a given antibiotic. And bacteria actually share this antimicrobial resistances quite frequently amongst themselves because keep in mind it's a question of survival for them. So if one of them acquires a method to resist an antimicrobial resistance, they share it with the other members of the microbiome. And how frequently and how easily that happens is studied quite extensively. And I'm showing here an example of research coming out of Heidelberg in Germany. So let's say an antibiotic resistance is acquired by a bacterium and it becomes resistant, here highlighted in red. It can either carry this resistance in its genome, here highlighted in this kind of greenish color, or even on a plasmid. So if this bacterium is close to another bacterium that is not resistant at this time, they can share the antimicrobial resistance by conjugation. They are forming this what's called a sex pili, which is a physical connection between those two bacteria, and it's forming like a tunnel where they can pass on the genetic information from cell A to cell B, and then afterwards both of them are carrying the resistance. And this can happen even between different types of bacteria. They don't both need to be of the same species or even same genus. It can be between gram positive, gram negative, between an E. coli and a Staphylococcus and a Pseudointermedius. They don't need to be even in the same phyla. It's a question of survival. I know I said it before, but there is a high pressure for these organisms to do and to acquire these resistances, so they will even pass this information on between different types. The second type is transformation. A bacterial cell leaks open, the genetic material becomes available to the environment, and DNA doesn't just lie around. It's a complex ecosystem, just like if a leaf falls in the forest, it will be degraded, it will be utilized, it will become nutrients for other trees. This is the same approach here for DNA as well. A different non-resistant bacteria might be able to take up this genetic material from the environment and thus becomes resistance as well. The third mechanism is by transduction using phages. Phages attack bacteria and infect them, and they jump from one to the next. So in this case, this phage is coming from this bacterium that was carrying the resistance, and it's ejecting the resistance gene into this bacterium that was previously uh, sensitive. Sorry, it was previously sensitive. So if you would do a simple thought experiment, let's say we have a dog suffering from atopic dermatitis. And for simplicity, let's say it's just 100 cells of Staphylococcus pseudointermedius causing the infection. And let's say one of them is carrying a resistance gene due to your mutation. If you submit this sample to culture and sensitivity testing, they will not test all 100 cells on your sample. It's simply not feasible. There's not enough space in the incubator. It's too much work for the technician. They will collect a few samples, and then they will collect a few colony forming units to do the sensitivity testing. So what are your chances that they will pick up this 1% of cells that are actually carrying the sensitivity uh, resistance? And so the chances are really low. So the chances are higher that the report will come back that the organism is sensitive, that the sample is sensitive. So you will start treating with the antibiotic. Best case, you wipe out all 99 of those sensitive ones, and the one resistance one will persist and now will flourish, will duplicate, and in 2 months the dog will be back with now a resistance infection. Otherwise, your one resistant organism can also pass on the resistance to other members and of the microbiome, and now you have an even more resistant infection. But by using molecular-based techniques, you target those genes. So it is much more sensitive in identifying even low copy numbers of these type of resistances. But again, you're using the genotype to predict the phenotype. But if this type of resistance gene is present in the microbiome, those bacteria will turn those machinery on and will use that genetic potential to survive the antibiotic treatment. But this is what I mean when I say that it can be more sensitive than culture insensitivity, because it is even detecting low copy numbers of these type of genes. And how this specifically applies in clinical diagnostics was nicely shown by this study. So now we're moving away from theory to real life experiences. They were, again, looking at dogs with atopic dermatitis, and they looked at the clinically healthy controls here and those with disease. So while I usually like showing donuts, they showed bar graphs to identify the bacterial profile. And in this case, every bar graph going up is an individual dog, and every different color you see is a different type of bacterial genus. So a donut is always 100%. Here, 100% is on the top, and you will still see a lot of white. That means that there were so many different genera that were in such little amounts. Remember how little my donut slices were, that they didn't show it to simplify the graph. So they only showed the most abundant different bacterial genus. And we can see here that Staphylococcus, the kind of salmon pink colored one, was present in almost all of the dogs, and some of them even at almost 90%. That's maybe one or two dogs, but the majority about 5 to 10%. However, in those dogs suffering from atopic dermatitis, overall there was a lot more Staphylococcus. Makes sense. It was probably Staphylococcus intermedius causing the infection in these cases. Then they started treating with antibiotics, and at the visit 2 did a follow-up and looked at the microbiome again. And the dogs are ordered in the same way. So dog number 1 here at the beginning had almost 100% complete overrepresentation of just Staphylococcus. After the treatment, it went down to almost 55, about 60%. And we now see here a Streptococcus coming up. But overall, the abundance of Staphylococcus went down, so suggesting that the antibiotic was efficiently targeting this pathogen. But you can also see Corynebacterium was overtaking in this particular dog, also another pathogen that could potentially be causing then a secondary other type of infection. So what this shows is that you can see in real time, is your antibiotic that you selected targeting the pathogen that you were interested in, or is it maybe wiping out commensals, which you want to keep? Or even if it's targeting the pathogen you were interested in, is maybe another secondary pathogen now coming up that could be causing a secondary infection that you now might have to worry about or not? Or do you need to potentially add a probiotic to restore the commensals? Or after an antibiotic treatment, after a washout phase, did I restore the commensals? Am I back to a clinically healthy, diverse skin that resembles more what we would see in a healthy control? So it offers you real-time patient management, and it offers real-time response to an antibiotic treatment. So even if the dog is already on antibiotics, you can see, is the microbiome resistant to it? What other pathogens might be coming up that I might not want to, but have to worry about later on. I briefly touched on biofilms at the beginning, and so to go in a little bit more detail here, According to the NIH, biofilms cause about 80% of all infections, yet they remain completely undetected by culture methods. So, how is that? Biofilms start usually relatively innocent. You have these planktonic bacterial cells that adhere to a surface, sometimes usually a cut, a little open wound, and those are the ones that are culturable in the lab. But once they start forming these microcolonies and the biofilm starts maturing, those biofilms become more diverse. They attract a large variety of different organisms that start proliferating in this type of environment, and then it starts maturing and growing over time. You start having this extracellular matrix that is a lot of different protein or even DNA, and it starts filming something that I like to refer to as a mega city with all its diversity. So you have the smaller houses in the center and then the skyscrapers, smaller houses on the outside, and then skyscrapers in the center. That's how diverse a biofilm is as well, and they have very different growth requirements. On the outside, you have more oxygen and more easy to digest sugars, which are eaten up by those bacteria here. But once you get into the center, oxygen is degraded, the simple sugars are all eaten up, so the ones in the center are left over with the more complex, more difficult to digest carbohydrates. And that generates a large diversity of different organisms. And these organisms, they communicate with one another. It's called quorum sensing. And it's their way of communicating, hey, there is an antibiotic targeting this structure. So it can even activate those organisms that are in the deep center of the biofilm to produce enzymes that break down the antibiotic or that reduce the pH of the overall biofilm so the antibiotic becomes ineffective and things like that. And because it is such a large diversity of different organisms, they have a large diversity of genetic potential, which makes it so difficult to treat them with antibiotics. But NGS can identify and quantify all those microbes inside the biofilm. You take this extracellular matrix and you put it in a test tube and you beat it. You physically break it up. You beat the hell out of it, like 20 minutes on a spot, quick beater, and it will break open not only the extracellular matrix, but also the cells that are present inside of it. You get the DNA out, and that way you identify who's there, how much is there, and what are they resistant to. So you have a better understanding of all the organisms and the genetic potential of antimicrobial resistances. And that's how NGS can actually help treat biofilm infections. Okay, well, Kitty asks, how come NGS diagnostics has not been used for years already? Overall, it helps with pathogen identification and treatment decisions. How come it's not widely used in clinical diagnostics already? The short answer is it was the cost. It would cost about $1,000 to test one sample. The turnaround time would be usually between 2 to 3 weeks in order for the runs of the sequencers to be full, to be in somewhat economically feasible. And that is simply too much time wasted, and it's not economical for veterinary medicine, especially. After that, the workflow is quite complex and requires a lot of quality controls and a very specialized lab. I briefly touched on it at the beginning, but you have to conserve the sample on the way to transit. You have to have an unbiased DNA extraction, no matter if it's a gram positive, gram negative, a spore. Um, a Malassezia cell is about 1,000 times larger than an E. coli cell. So you have to have an unbiased lysis, even if the cells are such different sizes. Then the PCR prep, the library preparation itself, the sequencing process, the bioinformatic analysis. You need to have a very good database that not only identifies which species is it, but also is it a commensal, is it a contaminant, is it a clinically relevant organism, what are the antimicrobial resistances, how to link the antimicrobial resistances back to the organisms. So it's a lot of artificial learning involved. And then finally, how do you digest all that information so that the veterinarian is not bombarded with 300 different organisms, but only the most clinically relevant organisms. So at this time, MiDOG is still the only clinical diagnostic lab that is offering this type of testing that helps even with chronic infections, as it covers all bacteria, all aerobes, all anaerobes, all fungi and 45 different antibiotics at this time with a turnaround time of 48 hours. So 2 to 4 business days, you do get the results, and it has the high sensitivity and sensitivity and allows for tailored antimicrobial therapy. So you do not have to worry about those tough to identify and tough to diagnose organisms like Mycobacterium, Mycoplasma, or Fusarium. You get all of those, but you also get the other ones that you did not have on your radar. The MiDOG report is consisting of 3 parts. First, pathogens, then antimicrobial resistances, then on the last page, commensals or any other additional information that might be important for your specific patient. And this is what the report would look like. Here I have an example of a canine otitis patient, and we would report on the clinically relevant microbes on page 1 for both bacterial and fungal pathogens. And all these are clickable links. Everything that's in blue, you can just click on and will take you to a search engine in case you want to learn more about those specific organisms. We highlight how much percentage of the overall bacterial profile are represented by the specific organisms. So in this case, the donut is all bacteria, but you can see right away Pseudomonas aeruginosa and Streptococcus canis are the main culprits causing this infection. And here you have the cell counts, just like you would see for culture results. And if it is a canine patient, we do have a clinically normal range as well. So we would find, even in a clinically healthy individual, a few cells of Pseudomonas aeruginosa, but just a few hundred. So that doesn't always warrant a treatment. So in this case, finding millions, that is way outside the clinical normal range. If this was a feline patient, you would still see millions of cells versus just 32 of of Aspergillus in this case. So you would still see the the clinically relevant pathogens right away. Here for the fungal profile, we do see a lot of Malassezia with just a few Aspergillus, and this is a great example of how important it is to have the absolute quantification. If you had submitted this sample for PCR testing and assume you have selected Malassezia and Aspergillus, both of them would come back positive, but Malassezia is 19,000 cells versus Aspergillus is only 32 cells. So the clinical significance would not be shown if it's just PCR both positive. You do need to have the cell count per sample. The first donut over here highlights, are you dealing more with a bacterial or fungal infection? In this case, more than 99% of all the microorganisms present are bacteria, so you would focus your treatment on these 2 pathogens rather than adding an antifungal. If this donut was 50 or 80% fungi, you would see right away on page 1, oh, I might need to consider an antifungal for this specific patients. So once you start using these results, these donuts are a clear and nice immediate indicator, oh, this is the kind of infection I'm dealing with. On page 2, we have the antimicrobial resistance profile. Here on top are the bacterial pathogens we identified on page 1, along with 45 different antibiotics. They are sorted by drug tiers, first, second, and third, to aid good antibiotic stewardship. So whenever possible, we would advise to select an antimicrobial from the first drug tier. And here is zoomed in how to read it. NR in red would be not recommended. And there's 3 different markers that go into not recommended. It's either we detected an intrinsic resistant, so that means the antibiotic is just off target. Remember the cell wall example that I brought earlier? Or we detected one of the antimicrobial resistance genes, and we're targeting about 100 genes at this time. Or the third marker is that it's less than 10% effective according to antibiogram studies. If none of those 3 are true, then the microbial would be sensitive. So we would have no resistance detected. Or if antibiogram data is available, we would give you additional guidance in good, fair, and poor performance of the antibiotic. This is kind of similar to the Target app. Now, there are some organisms like Pseudomonas aeruginosa or Streptococcus canis that there is a lot of antibiogram data available. Those are organisms that are very well studied. So in those cases, we can provide the antibiogram guidance. But an organism like Corynebacterium It does not grow well in the lab. There is no antibiogram data available. So in those cases, we do not provide this additional guidance and we just say no antibiotic resistance detected. So just keep in mind everything that's white and green and yellow is sensitive. As briefly mentioned on the example of canine atopic dermatitis, this allows for real-time patient management and antimicrobial therapy. You will see even resistances of those organisms that are in low abundances and thus aid good antibiotic stewardship. And our work has, and our antimicrobial resistance panel has been published in a peer-reviewed publication here with the example of cats from suffering from stomatitis. On page 3, we have additional or supplemental information, or what would be referred to in the culture report as commensals. We list all the top abundant organisms, both for bacteria and for fungi. And for dogs, we do have the clinically healthy reference range, which is shown here. And this is very nice to go over with your patients too, because it's colorful and I don't know, kind of fun to look at. And you can easily explain this is how diverse we would like your dog's ear to be. These are the two pathogens dominating your dog right now. This is why we need to use the antibiotic. And this is why it's important to actually follow the whole course of the antibiotic so we can reestablish this genetic and bacterial diversity. Again, for you, all of these are clickable links, so you can learn about each of them. Those numbers in brackets are the literature citations on why this is a pathogen. And then the same down here for the fungal profile as well. Again, we only have the clinically healthy range for dogs at this time, but we're actively working on adding feline and a lot of exotic patients as well. So despite the name, MiDOG is not just for dogs. We see all kinds of samples, all kinds of animals, and it can be used for swabs of the skin or ear or feces or urine. We do offer these different type of collection kits, and there is no cold chain required. As soon as you mix your sample with a DNA preservation agent that we provide either in those white or yellow tubes, the sample is stable at room temperature. It's safe for you to handle because it kills all bacteria, all viruses, all fungi in the sample, and you simply drop it in the mail and get it shipped. And with that, you then don't have to worry about those pathogens. Nothing will grow, nothing will decay during transit. It will be a true snapshot of how the pathogens were present in your patient. We do have a couple of success stories. They are also listed on our website if you do want to learn more. It's just to highlight that in ear infections in feline patients, we do quite frequently. It's not just a bacterial, but it's also a bacterial and fungal infection. Daisy was one of our patients that had a UTI for several years. For exotics, especially our exotic clients, are so excited because they're not limited to PCR testing anymore, which was kind of their standard because culture had all those shortcomings. We see a lot of yellow fungal diseases in reptiles and reliably detect even those pathogens in exotic patients. Here's an example of Dusty that was suffering from a chronic oral cavity infection, and one of those cases where I've rarely seen that lack of microbial diversity. Only two bacterial pathogens were dominating her sample, and the Klebsiella was carrying a lot of resistances, and she had been on amoxicillin before, and then based on this, her clinician was able to change the antibiotic treatment. I think it was to marbrofloxacin to treat both of these pathogens, and it finally cleared. Daisy had been suffering from urinary infections since 2016. Her owner did agree to culture testing repeatedly. It either came back negative or with Staphylococcus pseudointermedius. So her vet was treating her based on that. And then we finally were able to get a sample from her that was both submitted for culture testing and MiDOG at the same time. Culture that time came back with an Enterococcus fecalis versus MiDOG came back with Staphylococcus pseudointermedius, ureaplasma, and Mycoplasma canis. And based on those ureaplasma and mycoplasma, her vet then changed the antibiotics to marbrofloxacin, and she has been clear since then. And that was in 2019. We still follow her case, and she had not had a recurring UTI since then. And even though Micrococcus fecalis was detected in the isolate, in the culture isolate, we actually detected Enterococcus faecium, so a different type of species within the same genus. So again, it just shows the DNA doesn't lie. Culture and sensitivity relies on morphology of the cells, gram staining and things like that. But we use the actual DNA profile of those bacteria to reliably identify them down to the species level. Okay, so briefly to summarize the highlights of today's webinar, culture easily misses a lot of fungal infections. In our specific study, 97% of fungal infections and even 65% of bacterial infections. And thus, NGS has a higher sensitivity to detect and quantify those pathogens, even if they are unculturable or tough to grow in the lab, or if they would take several weeks to grow. And this is how NGS is more comprehensive than PCR testing. It's not limited to a panel. It's more sensitive than culture in detecting antimicrobial resistances. It has highlighted the importance of commensals and even at what point a commensal can turn pathogenic, and it allows for novel pathogen discovery. And with that, I'd just like to briefly remind you, microbes are everywhere. There's 100 trillion on each of every one of us, but using these molecular-based technologies, we can finally start identifying them. We can start learning about them, how they influence health and disease, not only for exotics, but also for our traditional pets. And with that, I'm at the end of my presentation. Thank you for your time. If you have any questions or comments, I'll be happy to answer them now. You will find more info about us on our website. You can also email me directly. And if you would be interested in any order information, I would refer you to Midwest Lab. Uh, the information is down here. Thank you. Do we have
Thank you.
any questions?
Thank you.
Hey, is there any in the chat? Nope. All right. So no questions. I take that as a good sign. If any other ones should come up later on, please feel free to reach out anytime. And then, yeah, I thank you for your time and thank you for joining us today.
Thank you.
Bye bye.